First, we have an atoms comes in a variety called elements. Some examples include oxygen, hydrogen, carbon, and so on. When you have more than one atom, that's called a molecule. So in this top row, putting a bunch of atoms together, you have molecule. Like for the first top row, you have 2 atoms each. Down here, these molecules, you have at least 3 atoms. Some molecule can be considered as compound. Compound is when you have more than one type of element. This first top row, although molecule, more than one atom, but not more than one element, so therefore they are not compound. For example, H2, there is only one type of element, which is H, and O2, there's only one type, which is O. This row are both molecule and compound. For example, NO, you have 2 atoms, but at the same time you also have 2 type of element, the N and the O. And H2O, you have 3 atoms and you have 2 elements, H and O. And the rest that's on the bottom, they're all molecule at the same time. They're also compound, except for C2. There are many molecules, but the most important molecule that is very diverse is water. That's why there is a lot of studies on water. So here are the properties of water. Cohesion and adhesion. Because of the hydrogen bond found in water, which account for the cohesion, therefore water bonds to itself. Adhesion is when water bonds to other. The result of water droplet is from cohesion, water bonding to itself. And when these droplets sticks to other stuff like plants, that's adhesion. Cohesion and adhesion work together with transpiration to pull the water up the tree, which is very important for plants. The hydrogen bond in water also accounts for the high specific heat. The bond can absorb a lot of energy before it's broken off. Therefore, it can absorb a lot of heat. Absorbing a lot of heat is important because in coastal area, it keeps the land warm. At night, it also keeps the land warm by giving back the heat to the land. Water also has heat of vaporization, which means it can turn from a liquid into a gas after a while. This is important because, for example, like when you sweat, you release the hot water in your body, and the hot water is carried away by the wind, and that cools you down. Another important property of water is that ice float. So when water turns into a solid, the ice float. This is important because if ice were to sink, then it would crush all the fish or freeze all the fish, and the fish would die, which leads to extinction of human as well. Also, when ice float, it creates like a blanket layer at the top of the pond or the lake, and it insulates the bottom layer, so then the animals or the fish stays warm year-round during the winter time. Another important property of water is that it's a universal solvent. Water is able to dissolve many things, the solute. Besides the fact that it makes our drink tastier, our body is full of water. And then everything that we absorb into our body, food for example, we have to break it down to smaller pieces. That way our body can digest it. That's why water as a universal solvent, dissolving these molecules is important. Because when water dissolves the molecule into smaller parts, then it can be absorbed into our cell. Let's go back to our list. Now, when you have a bunch of molecules put together and you have bigger molecule, you have what is called macromolecule. Macromolecules, there are 4 of them, and they are the basic building block of cell. The 4 macromolecules are carbohydrate, lipids, proteins, and nucleic acids. Each of these has 2 categories. One is the monomer version, which is 1 part, and the other one is the polymer version, which is the many part. The monomer version for carbohydrate sugar is glucose and ribose. There are many other, but these are the 2 most important ones. And then lipids, there is glycerol and fatty acid. Under protein, there's amino acid, and under nucleic acid, there's nucleotide. It's important to be able to recognize these structure because you will see them on many tests. Carbohydrate is recognized by the ring formation. Glucose has a 6 ring which forms a hexagon, and ribose is a 5 ring which forms a pentagon. Under lipids, the glycerol is the purple part, and the fatty acid is the long yellow part, which is a hydrocarbon. The amino acid is easy to recognize by the amino group, which has the N in it. And nucleic acid, the nucleotide, is also easy to recognize because it has the phosphate group. If elements were actually under this category, then a monomer of elements would be the atom, and then a bunch of atoms put together forming a polymer, you have molecule. Okay, now let's list the polymers of the macromolecules. Under carbohydrate, you have polysaccharide, and their example are starch, glycogen, cellulose, chitin. Under lipids, you have phospholipids and steroid. Under protein, there's polypeptide, and under nucleic acid, you have polynucleotide. Now let's take a look back at elements. How does an atom attach to another atom and become a molecule? It's attached to another atom via bonding. There is two types, ionic and covalent. Same thing with these macromolecules. They too have a bond, but they just have different name. Carbohydrate, we have glycosidic linkage. Glycosidic is another word for glucose, therefore it's just another word for carbohydrate. Sugar linkage. Under lipids, you have ester linkage. Under protein, you have polypeptide bond, and under nucleic acids, you have phosphodiester linkage. The ester linkage and the phosphodiester linkage is not as important, so don't worry too much about those. And that's how you form from a monomer into a polymer, the thing that's in the middle between those two. Now let's go back to our polymer for a macromolecule. We're going to start with carbohydrate. Under carbohydrate polymer, you have a polysaccharide. Two examples of polysaccharide is starch and glycogen. Starch and glycogen are both sugar, and then starch is found in plants, sugar in plants, whereas glycogen, it's the sugar found in animals. Both of these are digestible, and their linkage is actually an alpha linkage. Notice that the middle between them, it's an alpha linkage, meaning it's just pointed down. You'll see it later compared to a beta linkage and how it's different. So these polysaccharide polymer is just simply a bunch of glucose stuck together via an alpha linkage, and they're digestible. Two other polysaccharides are cellulose and chitin. These are beta linkage, and therefore they're not digestible. So take a look at the picture of our molecule, and you notice that the linkage, the O in between them, it's up and down, it's not both all the way down. This is beta linkage. And for this, just that small slight differences, that's why they're not digestible. Cellulose is found in cell wall, and chitin is found in animal, like usually arthropods, their exoskeleton. Cellulose, because the fact that why can't we digest plants, then what's the point of eating plant, is because it helps us clean our inside. The cellulose will go into our body as fibers, and then it'll come out pretty much the same thing, and because it's not digestible, it's designed simply to clean your inside. Next, we're going to move on to triglycerides under lipids, which has the two polymer, phospholipids and steroids. The polymer for lipid is triglycerol, which is when you have the fatty acid bind to a glycerol and you have three of it, then that's why it's called a triglycerol. Two examples are phospholipids and steroids. Phospholipids are found in the cell membrane. Both of these, as lipids, they act as stored energy. And phospholipids, it's known for the famous hydrocarbon, which is a very long chain of carbon and hydrogen together, which is the tail. That's the hydrocarbon. The hydrocarbon, the tail, is non-polar, so therefore it's against water. So it is hydrophobic, which means it doesn't like water. That's why in the cell membrane at the bottom, notice that the tail is found on the inside, whereas the head is hydrophilic. They're polar, so they like water. That's why they're on the outside. Next, we move to proteins polymer called polypeptide. Polypeptide for proteins, two example is proteins. Yay, good. It's called the same name as its original. That makes it easy. And the other one is enzyme. The purpose for proteins are listed as follows: structural, storage, transport, hormonal, receptor, and defensive. These are not as important as much, but they are still there. However, the most important one is enzymatic proteins, which it acts as a catalyst to speed up the chemical reaction in your body. Now, protein is found in 4 structures. First is a primary, which is simply just amino acid as a chain. Then you have secondary, where those amino acid is then folded into two-way, either circular like spinning stuff called alpha helix, and then the other one is beta pleated, which is like a fold. That's the one, the picture on the bottom. And then those, when you put them together, you get an even longer chain, which is tertiary, and an even longer chain. When you have a clump, you have a bunch of clump put together. That's called quaternary. And some of the most famous quaternary protein that we need to know is hemoglobin and collagen, which you will learn later on. Note that protein is opposite of carbohydrate and lipids, because protein, you need to put them into the structure in order to work, whereas carbohydrate and lipids, you need to break them down in order to use them for energy. So protein, if you break them down, they no longer work. They get denatured. And two ways to break them down is too much pH or too much temperature, either too high or too low, one of those two. So protein, opposite of the other two, is the other two you must break them down. Protein, you shouldn't break them down, because if you break them down, it wouldn't work. You have to keep them in their structure. Okay, yay, we're on our last one. Nucleic acid, the polymer is polynucleotide. Example is simply RNA and DNA. We're not going to go into detail about here, because later on there's going to be a full section about DNA and RNA, which is all the detail you need. So for now, you just know that DNA is the one on the left side, double-stranded, and RNA the one on the right side, single-stranded. And that's pretty much it for the review. Hope that helps you guys. Good luck on all your tests.